Welcome everyone. I'm Dr. Kelly Deal, and today I'll be describing the new studies sponsored by the Golden Retriever Foundation as of January 2026. We're very excited to share several important projects that build on previous work and open new directions for improving canine health. The first study I'd like to cover is a continuation of an ongoing project also funded by the Golden Retriever Foundation. This work is coming from the Animal Medical Center and is led by Dr. Alexander LaRue. The study focuses on targeting the integrin alpha 6 beta 4 complex in hemangiosarcoma. In the first phase of this study, Dr. LaRue examined hemangiosarcoma tissue to identify a cell surface marker that distinguishes cancerous tissue from normal tissue. He identified a molecule called integrin alpha 6 beta 4. While the name is complex, what's important is that this marker appears to be highly specific for canine hemangiosarcoma. This new project has two main goals. First, the team will study samples from 50 dogs with hemangiosarcoma. They want to see how the presence of the integrin alpha 6 beta 4 relates to survival, what genetic changes happen in the cancer when this integrin is present, and how it possibly helps cancer cells grow and spread. Second, the team will design canine antibodies that can target the integrin alpha 6 beta 4. They will create two kinds of treatments with these antibodies, one with radioactive particles and one with chemotherapy drugs. Both will aim to kill cancer cells and should cause less harm to healthy tissues than current treatments. This could represent an important step toward developing a highly targeted and specific treatment for hemangiosarcoma. Our next study comes to us from CNRS in France and is led by Dr. Benoit Hedon. The study is titled Genetic Investigation of Histiocytic Sarcoma in Golden Retrievers, Identifying Predisposing Variants Through Whole Genome Sequencing. As many of you may be aware, we are seeing a high proportion of histiocytic sarcoma in dogs enrolled in the Golden Retriever Lifetime Study. Dr. Hedon will compare the genomes of dogs diagnosed with histiocytic sarcoma to healthy age-matched control dogs. His goal is to identify genetic variants that may predispose dogs to this devastating cancer. Identifying genetic risk factors could be an important step toward developing a diagnostic test. Knowing whether a dog is genetically predisposed would be extremely valuable for breeders, veterinarians, and owners. Next, Dr. Cynthia Otto from the University of Pennsylvania is expanding on her previous work with Morris Animal Foundation. Her study uses a combined sensor approach to examine volatile organic compounds, or VOCs, as potential markers of canine hemangiosarcoma. Dr. Otto is well known for her work training dogs to detect specific scents. She has been using samples from the Golden Retriever Lifetime Study to determine whether trained detection dogs can distinguish, using just a few drops of blood, between dogs with hemangiosarcoma and those without. She found that trained biodetection dogs have been able to correctly identify canine hemangiosarcoma blood serum samples from lineups which include serum from healthy dogs and dogs unwell with other conditions, with an average accuracy of 71%. Building on this finding, the proposed study will chemically analyze the VOCs in the blood of dogs with confirmed hemangiosarcoma, healthy dogs, and dogs with other illnesses. These VOCs will be recreated as synthetic odors and presented to the trained dogs to help confirm which parts of the odor are most important for detecting the disease. While the team does not anticipate the biodetection dogs as part of the final diagnostic test, they serve as critical biosensors in this research. Their ability to detect specific odors present in hemangiosarcoma will help us define the VOC signature of this disease. Our next study comes from Virginia Tech and is led by Dr. Ryan Singer. The study is titled Detection and Evaluation of Treatment Efficacy of Visceral Hemangiosarcoma Using Raman Molecular Urinalysis Fingerprinting. Wow. This project also focuses obviously on hemangiosarcoma. Dr. Sanger has developed a urine-based testing method called Raman Molecular Urinalysis, or RMU. He will evaluate whether this technique can distinguish between dogs with hemangiosarcoma, dogs with cancers other than hemangiosarcoma, and other diseases. Because urine is relatively easy for owners to collect at home, this approach could have really important implications for early diagnosis. It may also be useful for monitoring treatment response and tracking disease progression or recurrence over time. Finally, our last study is led by Dr. Andrea Beal, a veterinarian at EpiPause. Her project focuses on characterizing genome-wide DNA methylation profiles in dogs diagnosed with obesity and age-related diseases. Dr. Beal will compare Golden Retriever lifetime study dogs with and without obesity, as well as dogs with and without age-related conditions. An example would be osteoarthritis. We know that aging causes changes inside all the cells of the body. These DNA biomarkers can be reliably linked to one's age and health, and they differ between individuals depending on their health. In this pilot study, the research team will search for DNA biomarkers that may play a role in aging and disease. They'll compare healthy dogs to dogs that are obese or have an age-related disease, and they're using samples from the Golden Retriever lifetime study. The goal is to identify new markers to explore in future studies for their potential use in early disease diagnostics, treatments, and maybe even prevention strategies. Thank you for listening to this overview of our new studies. On behalf of Morris Animal Foundation, we are deeply grateful to the Golden Retriever Foundation for their long-standing partnership and support. We are very excited about these new studies and the impact they may have on improving the health of golden retrievers and dogs everywhere.